Has the disease mechanism of COVID-19 finally been unraveled? It's possible. Hello everyone, this is Dr. Chris Martinson with your update on SARS-CoV-2. It's September 3rd, 2020. Today we're going to be discussing the Bradykinin storm, or why vitamin D and Ivermectin seem to work, along with some other things we've been recommending over time. Let's take a quick peek, and by the way, this mechanism is a little bit complicated. It's so worth understanding, and it actually makes this virus worthy of the honey badger nomenclature because, oh my gosh, it's just astonishing if this hypothesis is correct. This virus is a lot more complicated than I thought at first. And I know they're all complicated because guess what? Hijacking cell machinery is not an easy thing to do, and you've got to have a pretty sophisticated way of getting in there and unlocking a super complicated system. Well, that's what viruses do. At any rate, thank you all for this suggestion. I just got a lot of people coming in saying the same thing. This is from Vladimir saying, very interesting, maybe not a cytokine storm after all, or at least not the main culprit. And I'm going to agree with Vladimir here. I think the Bradykinin storm that we're going to talk about is part of it. I don't think it's all of it, but, it's hypothesis. We'll see. But it could be maybe the main culprit. Love the information you give us and the tools and lessons for how we can dig in ourselves. Thanks again for that too, Vladimir, because that's really what I'm all about here. I'm trying to arm people with the information and as well a way of going about and getting that information. Because what I do is I have a way of looking at this stuff, which is independent, skeptical. I don't have to impress any colleagues here. I don't have a chair of a department who's going to frown upon me if I say things in a certain way, and I don't have any pharma companies looking over my shoulder telling me what I should think or do. And in addition to Vladimir, Thomas came along with this and said, please, can you look at this? David said, yeah, thought this was interesting, and this is the article here. And hey, more people too, all these other people, Pete and Stolen Fates here, Sequoia, Bruce, and even some more. Lots of people weighing in saying, hey, could you please take a look at this? So happy to. Let's dive in. And before we dive into the actual article, let me remind everybody, way back in April 23rd, right? I put this slide up here, and part 4, which we kind of haven't focused on in a while, is I said, just, okay, you got to treat everybody, but then collect the data, and then you got to share it. And what I said back there on April 23rd was I said, listen, if the United States, if I was in charge, tens of billions of dollars just run clinical trials, right? And I would redirect the FDA and the CDC to become massive data collectors for every clinical trial. And then you just test everything, right? Everything you can think of. Just try it. Just try it. Chinese herbs, hydroxychloroquine, remdesivir, every antiviral known, plasma therapy, monoclonal antibodies, everything. Anything that you find in the literature that says this thing had antiviral properties, give it a go. To which we have to add ivermectin into that list, which I didn't know about it as strongly on April 23rd as I do now. But then you got to collect massive amounts of data. You got to anonymize it, maybe even make it public, announce a huge prize for anybody that can crack the code, and the X Prize story where some people came forward and put up $10 million to see if they could spur private space exploration. At the time, everybody thought that was nuts. NASA has a lock on it. You need to be a government to do this. Well, guess what? Not true. And now we have private exploration because of that X Prize story that came along. Anyway, that's what I thought would make a lot of sense. So guess what? Now we have somebody came along and took some data and crunched it through some supercomputers and came up with this really interesting hypothesis. Now, it's set up to that hypothesis so we can understand a little better. Regulating the blood pressure in your body is actually a super complicated thing. It might sound easy. You want to have 120 over 80, diastolic, systolic pressure. You have this blood pressure, right? But it's really complicated how that's regulated. There are 5 main factors regulating blood pressure, which is the output of your heart, the peripheral vascular resistance, like how pinched down are those pipes at the end, because your heart has a very big aorta that pumps a large volume of blood out, but that goes all the way down to the finest capillaries in your most distant toes and squeezes through those capillaries and then comes back up your veins. So looking at the amount of resistance in that overall process obviously can influence blood pressure. The more the resistance, the higher the blood pressure. The higher the volume of circulating blood, the higher the blood pressure. Also, I should have said, I guess, the higher the cardiac output. The more viscous your blood, the higher the blood pressure, and the less elastic your vessel walls are, or the more rigid they are, the higher the blood pressure. And then the reverse of all of this, right? If you want to have lower blood pressure, reduced cardiac output, reduced vascular resistance, reduced volume of circulating blood, reduced viscosity, and increased elasticity, those would be all things that would contribute to reducing blood pressure. Now, sounds simple, but oh my God, is it complicated how the body goes about doing that. And by the way, it's a complex system. Your body's never just doing one thing at any one time. It's doing lots of different things. And many of the factors that influence blood pressure are also influencing other things like your immune functions, and whether or not there's fibrosis and cell remodeling going on, or programmed cell death, all that stuff. How complex is it? Let's just look at one tiny piece. This will all be on the test tomorrow. Okay, moving on. No, just kidding. Let me back up. When we look here, this is mostly looking at the RAS system, RAS, the Renin-Angiotensin System. And so angiotensinogen is this big, large protein, large-ish, that then gets cleaved by various proteases, and it gets turned into all these products. And as it comes down various pathways, so we might say it comes down here and goes across here and comes down here. Well, if it came down this pathway, it would lead to vasodilation, dilatation. So that vasodilatation makes your blood vessels open or expand. That would reduce blood pressure because it's going to decrease the peripheral vascular resistance. So anything that's vasodilatation here is a vasodilator. These things are all going to reduce blood pressure, but what's the opposite of that? The vasoconstriction right here. So again, noting, say, angiotensin 1 coming down, turning into angiotensin 2, binds to this receptor, the AT1R. It would lead to vasoconstriction, cell proliferation, fluid volume changes and homeostasis, fibrosis, all this stuff, right? So this body is doing a lot of very complex things with multifunctional factors and receptors. So lots of stuff happening. You don't have to understand any of this to understand the Bradykinin hypothesis, not at this level of detail, but I just wanted to point out lots of cascades, lots of things running around to roughly help regulate the blood pressure and do a whole bunch of other stuff. Regulating apoptosis, which is programmed cell death, influencing neuronal development, controlling inflammation, all of these things. So a lot of complex things. So what is this hypothesis? Well, this is really cool, really nice piece of work. And I just loved what they came up with here. This is the paper, a mechanistic model and therapeutic interventions that are recommended for COVID-19 involving a RAS-mediated bradykinin storm. So that's the Renin-Angiotensin System, which is this whole thing. But now they're focusing on this. It just looks like an just one little piece over here. There's a huge story around this pathway right here. All right. And we're going to decode a lot of these words right here. This is the main abstract. And there was some really good stuff in here. I want to just take it step by step. Then we'll go into the explanation. And this fits with a lot of things, and it really points towards and validates a lot of the recommendations I've been giving around some of the outpatient treatments that we could all take, including zinc, vitamin D, all that stuff. So let's dive in here because it's exciting, super exciting. The entry point for the virus, the coronavirus that we've all been struggling with, is ACE2. ACE2 is the receptor. That's the angiotensin converting enzyme 2, ACE2 receptor, which is a component of the counteracting hypotensive axis of RAS. So the hypotensive under, so that's leading to lowered blood pressure. Bradykinin is a potent part of the vasopressor system that induces hypotension and vasodilation and is degraded by ACE, not ACE2, ACE. That's ACE and ACE2 are two totally separate enzymes here, okay? ACE and ACE2. So it's degraded by ACE and is enhanced by angiotensin 1-9, which is produced by ACE2. Don't worry, I'm going to give you pictures around all this. We're just going to go through the language right now. We'll decode it all in just a second. So what they did here is they performed an analysis on gene expression data from cells that they got from bronchoalveolar lavage fluid, BALF. That bronchoalveolar lavage fluid was washed out of the lungs of COVID patients. In that fluid, there's all kinds of stuff, but cells from the patients. They took those cells and then they looked at the gene expression that was going on in those cells, and then they compared those against healthy controls. All right. So comparison with BALF from controls, they identified a critical imbalance in the RAS, Renin-Angiotensin System, represented by decreased expression of ACE, this one up here, this ACE receptor, in combination with increases in, not just, get this, not just one, not just increases in ACE2, but also increases in renin, angiotensin, KRAS receptors, kinogen, many kallikrein enzymes that activate it, and both bradykinin receptors. So a downregulation or a decreased expression of ACE itself and an upregulation in everything else. Now, what a honey badger of a virus, right? It's not just content to come in and hijack some of the cell machinery and make a few extra copies of itself. It's also going in deep into the cell, monkeying around inside the nucleus in order to dial down some genes and their expression and dialing up other ones. And so it's not just the virus itself causing a direct impact into the cell and causing cell injury, which leads to all sorts of inflammation and things like that. It's going inside the cells and causing them to completely dysregulate which sort of products they're making, what sort of proteins they're making, which sort of receptors they're making, so that the virus's impact is greatly magnified on the host. All right, continuing on, they said this very atypical pattern of the RAS is predicted to elevate bradykinin levels in multiple tissues and systems that will likely cause increases in vascular dilation, vascular permeability, and hypotension, low blood pressure. So vascular dilation, all the blood vessels relax, they're smooth muscle cells, and they get big and open. Vascular permeability, so they get leaky, and all of that leads to hypotension because, as we mentioned before, anything that's going to decrease peripheral resistance and the elasticity of the vessel walls, those two things are combining to lower blood pressure. And also we have the leaky vessels, and that's going to actually decrease the volume of the circulating blood because a lot of the fluid in the blood is now leaking into the tissues and into the lungs instead of staying inside the pipes, the blood vessels. All right. So these bradykinin-driven outcomes explain many of the symptoms being observed in COVID-19. So the whole thing ties together really nice, really cool, and explains a lot of things. And the good news from this is it points to a bunch of different treatments we could try. It's a very testable hypothesis. It should be pretty quickly determined clinically whether this is an accurate one or an inaccurate one. So to me, that's science. Hey, they came up and they punched through just big data, and they put some supercomputers against this, crunched through literally billions of variables, and this is what popped out. So the authors described it as a real eureka moment. It's a real win for big computing and AI if this can really be borne out. So there's a whole beautiful explanation on Drbeen's site. I can't recommend it highly enough. He's a great teacher, great instructor, and got the link to it down there on this right here, down at the bottom here. And he talks about this paper, and he talks about the whole bradykinin pathway. I'm not going to repeat it and recreate it here because he's just done such a great job. I couldn't do anything but do a worse job at it. But the basics of it are that there are all these precursors out there. These are larger proteins that get clipped by this kallikrein, which is a protease. And it snips these and takes these bigger proteins, turns it into bradykinin. And then bradykinin comes and attaches to a receptor and releases things like nitric oxide and PGI2 and EDHF here, which is endothelial-derived hyperpolarization factor. Very complicated stuff, but just all these things end up leading to vasodilatation. So that vasodilation just opens the blood vessels up, and guess what? You get lower blood pressure, all that. But bradykinin could be the ACE receptor. ACE, not ACE2, but ACE itself sees the bradykinin, chews on it, and breaks it down into bradykinin 1,5, which is an inactivated form. As well, the ACE receptor over here takes angiotensin 1, converts it to angiotensin 2, and what does angiotensin 2 do? Well, that's one of the few things that leads to vasoconstriction, which can reverse the process of vasodilatation. So some things are making your blood pressure go down, some are making it go up. They're doing that by both making your blood vessels relax but tighten up. This is one of the few pathways that leads to vasoconstriction right here, which is the angiotensin 2 pathway. And then if angiotensin 2 is floating around, it runs into the ACE2 receptor, it then gets converted into angiotensin 1,7, which leads to vasodilatation. All right, so that's the basic pathway here. So if we look at this, to put it back in very simple terms, if you got a lot of angiotensin 2 running around, which runs through the ACE pathway, that's going to lead to that vasoconstriction. That's going to lead to this increase in blood pressure. On the other side, angiotensin 1,7, as converted by the ACE2 receptor, and as well as bradykinin, both of those are working to create lower blood pressure. So those two things are supposed to be kept in really tight control, one then the other, and really tight balance. And there's a bunch of other factors involved, very simplified here, but that's the core of the symptoms of the system, is having those things all in balance. So the pieces that want to drive blood pressure up and drive it down, and sometimes you need higher blood pressure, sometimes you can relax a little. If you're being chased by a bear, all these other things, you want to have a lot of blood pressure to go along with that, and increased heart rate. A lot of factors, very complicated, but at its heart, it's a very, very finely tuned system. And when something like COVID or SARS-2 comes along and it dysregulates that system, a lot of things can go haywire. So it's not maybe so much your body fighting itself as the virus coming in, being a super sneaky honey badger, somehow getting all the way into the nucleus and driving your gene expression to throw these components out of whack. That's the hypothesis here. So let's dive into it and take a quick look. All right, let me just get rid of that part right there because I don't think we need that. All right. This is in their paper here. They say when COVID-19 first emerged, a team of researchers in China studied fluid and cells collected from the lungs of patients to help them identify the SARS-CoV-2 virus. Here, Garvin et al. Analyzed the data collected in the previous work to investigate whether changes in how the body regulates blood pressure may contribute to life-threatening symptoms of COVID-19. The analyses found that SARS-CoV-2, the virus, caused the levels of ACE in the lungs to decrease, while the levels of ACE2 increased. So now we can see if you're decreasing this, well, you don't have this pathway anymore. That's gone. And if you're increasing this, you not only have more places for the virus to attach to and come in and start monkeying with cells, but you're going to get an increase in this as well. So it's blocking the vasoconstriction, and it's leading to the vasodilation part. All right, so let me get this on here. This, in turn, increased the levels of a molecule known as bradykinin in the cells, referred to as a bradykinin storm. Previous studies have shown that bradykinin induces pain and causes blood vessels to expand and become leaky, which will lead to swelling and inflammation of the surrounding tissue. Those are all classic symptoms of COVID, right? In addition, the analyses found that production of a substance called hyaluronic acid was increased, and the enzymes that could degrade it greatly decreased. Hyaluronic acid can absorb more than 1,000 times its own weight in water to form a hydrogel, and of course I posted those awful pictures way back in February of this goop that was being aspirated out of the lungs of the poor COVID patients back then before we knew how to treat them very well. People were being put in prone positions and just hung upside down, in essence, to allow this goopy stuff to get out of the lungs, right? The bradykinin storm induced leakage of fluid into the lungs combined with the excess hyaluronic acid would likely result in a jello-like substance that is preventing oxygen uptake and carbon dioxide release in the lungs of severely affected COVID-19 patients. Therefore, the findings of Garvin et al. Suggest that the bradykinin storm may be responsible for the more severe symptoms of COVID-19. So very quickly again, not to get overly complex, not doing this, but just so that we all have this orientation around this, the RAS, Renin-Angiotensin System, angiotensinogen is a precursor protein. Renin is an enzyme that comes along, it clips it, it turns it into a smaller protein, angiotensin 1. ACE2 can turn that into angiotensin 1-9. But if ACE itself here sees that angiotensin 1, it clips it, turns it into a shorter thing called angiotensin 2. Angiotensin 2 comes and binds to these two receptors, the AT1 and AT2 receptors. Okay? If ACE2 sees angiotensin 2, it can clip it, turn it into angiotensin 1-7. That can hit the MOS receptor over here. And as well, renin and prorenin over here can come into the prorenin receptor. So these are the components of that overall renin-angiotensin system, and they're all sort of feeding off of each other. And again, just as a reminder, a lot of things lead to this vasodilatation here. Vasodilatation. So that MOS receptor, if you have angiotensin 1-7 coming down the ACE2 pathway, and so ACE2 doop, 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 comes down here. It comes down into the MOS receptor, you get that vasodilatation, which you see coming down this pathway here. Same thing. And as well, what do you get? It's anti-inflammation, anti-fibrotic. All those are actually pretty good things right there. Lots of things lead to this vasodilatation, and really just this one pathway for the vasoconstriction, which requires ACE, the angiotensin converting enzyme. Not ACE2, but just ACE. So you have to come down this pathway here in order to get to that little spot of vasoconstriction. So check this out. This is what they actually found. So again, they washed cells out of the lungs of these patients. They took those cells. They compared their gene expression of those cells compared to normal cells. All right, ACE2 is also the main receptor for SARS-CoV-2 virus, not highly expressed in normal lung tissue. So how did the lungs get so badly hit? Based on the genotype tissue expression 6 population. So they've run these genotype tissue expressions on large populations and said, where are genes expressed, what receptors are seen in which tissues, all of that. However, they say here, continuing, results from our differential gene expression analysis of RAS genes in cells taken from BAL samples, those bronchial alveolar lavage samples from individuals presenting with severe symptoms of COVID-19, demonstrates upregulation of ACE2 199-fold and disruption of this system compared to controls. In the COVID-19 samples, AGT, a 34-fold increase in the enzyme that activates it, REN, 380-fold are increased compared to controls, whereas the enzymes that produce the most of the cleavage products, including ACE, a minus 8-fold decrease or downregulation, which will likely result in a shift of the entire RAS to produce angiotensin 1-9. Just shoves the whole thing down over to that one pathway. In addition, the AGTR1, 430-fold increase, and AGTR2, 177-fold increases in these receptors are upregulated in the BAL COVID-19 sample. So from their paper, this is going to take a second to walk through, but it's worth it. It's totally cool. Check this out. What they're showing here is these are the gene products, and they're measuring how much they are either upregulated in red or downregulated in blue. And this is a log base 2 scale. So a log base 2 of -5 or 12, very quickly, what does that mean? We're all familiar with log 10, right? It goes up from 1 to 10 to 100 to 1,000 to 10,000. Log base 2 takes us to something here where if we saw a 5-fold increase, log base 2 to the 5 gives you a 32x increase, 6 gives you a 64x increase, 4 gives you a 16x increase. So that's just to orient us around log base 2 again for a quick math reminder. So if we see something like a log base 12, wickedly upregulated, just that's a very, very large number. At any rate, what they're showing here is very cool. So let's take it one piece at a time. First, ACE heavily downregulated. We're seeing a lot less ACE. So we've got to cross all these ACEs out, right? They've just been taken out of the system. Somehow, COVID, as the disease, the SARS-CoV-2 virus comes in and messes around with the gene expression in the nucleus of the cell so that fewer gene products are made and more gene products of other things are made. What a complicated system. But at any rate, let's talk about what it is doing. It's whacking the ACE2 bits right here. And it's also whacking this thing, the VDR, which is part of the vitamin D pathway here. And that explains why if you have more vitamin D in your system, that can actually really be helpful because it's inhibiting some of this VDR, the vitamin D receptor, which, by the way, is a thing that helps to block renin. So if you don't have that going in, you don't inhibit the renin system. Now, check this out. What's upregulated really, really super strongly here? Well, ACE2, highly, highly upregulated here. AGTR2, highly upregulated. Lots of that stuff kicking around. AGT, lots of that kicking around. Lots of renin here, and so on. And so what does this mean? It means that we're really seeing one pathway highly, highly amplified, and the other pathway, which is countervailing pathway, really, really shut down or inhibited by the action of this particular virus. So what happens when you block the ACE portion, but you upregulate the ACE2 portion and the bradykinin side? Well, that's pretty simple. We're blocking preferentially this side of the story, and we're vastly increasing this side of the story. So everything in this story is pushing down the overall blood pressure and pushing down and increasing vascular leakiness and having things leak out of vessels and all of that. So really cool system there. And so when we look at this as well, one of the pieces that gets impacted by this is hyaluronic acid is part of this overall complex pathway. A lot more hyaluronic acid is, by their hypothesis, is predicted to be increased its production. And so if that happens, you're going to get this, a normal air sac with air coming in and a normal alveoli in the lung doing its nice thin gas permeable membrane thing is now blocked by having this big hydrogel that's sort of blobbing around in there. And you have all the inflammation and all sorts of things just coming along and basically ruining the lung function. So the question then becomes, well, if this COVID thing, if this SARS-CoV-2, if the honey badger virus is in there monkeying around, upregulating some gene expression and downregulating other gene expression, well, it's having an impact in the nucleus of the cell. So little cell biology reminder, every cell in your body is surrounded by a membrane. But inside of that, it's actually a double membrane system, because inside of that membrane, you go through some cytoplasm and you get down to the nucleus of your cell. The nucleus itself is surrounded by its own membrane. And each membrane, whether it's the cell membrane or the nuclear membrane, has a really important set of jobs, but their number one job is to keep a bunch of stuff out and keep other stuff in, right? That's their main job. So the cell membrane has a very different job than the nuclear membrane, but the nucleus is very, very carefully guarding what comes in and what comes out, and it has a set of proteins that are tasked with allowing things to come in and allowing things not to come in. And so the theory here works like this, that there are actual proteins called importans. There's important alpha and important beta, two subunits that come together, and those importans, when they are together, they have a binding site where something can get attached to that, and it gets taken through the nuclear membrane. So this is the cytoplasm. We're out here in the cytoplasm. This is the inside of the nucleus where all the DNA replication's happening and genes are being upregulated and downregulated. Well, this stuff has to get through this nuclear pore complex, which is this giant, very, very sophisticated protein complex that its job is to not allow anything to come through unless it's being shepherded through by the importans. And so the idea is that the coronavirus would attach to the important complex and get carried in, and then it somehow enhances viral replication, possibly, or It's getting in there and turning on some genes and turning off some other genes. And we don't have that mechanism worked out yet, but somehow it's upregulating things. The theory is that it needs to get into the nucleus somehow. Not maybe the whole coronavirus is depicted here, but certainly components of that, certain proteins of the coronavirus would get imported into the nuclear pore complex. And that's part of its life cycle and part of why it's a bad boy. All right. So ivermectin, kind of cool. Ivermectin actually binds to the important alpha subunit. And because it does that, it prevents the important complex from doing what it does. It prevents the important from bringing viral things into the nuclear center. That's how ivermectin actually works. That's its mechanism of action, at least part of it. And it's well known. This is from a paper that was hypothesizing about how ivermectin might work to help limit COVID as a disease. And we already know that ivermectin has really strong antiviral actions, right? We know that it's active against the Zika virus, influenza A, VEE, West Nile, we got Newcastle, HIV-1, yellow fever, dengue, Japanese encephalitis, tick-borne encephalitis. There's all these antiviral components that are already known about ivermectin, which is typically an antihelminthic, antiparasitic thing that's given to horses and dogs and cats and things like that. But it's a really, really cool compound. And it not only has these antibacterial and anti-cancer and endectoparasitide activities, but it's got all these antiviral things going on as well. So we know this. We've known this for a long time. We, the scientists, then people who are in this space. So it shouldn't have been that much of a surprise to find that ivermectin actually has some sort of a role against coronaviruses too, in this case, SARS-2. At any rate, here's the idea that it would, because ivermectin, shown here, is binding to the important alpha subunit, it prevents this dimer from binding and then entering into the nucleus. So that's how ivermectin would work in this particular story, and there's strong reason to suspect it would because, hey, guess what? It works against a whole lot of other viruses through this sort of a mechanism. So it's not that much of a stretch to suggest it's happening here. By the way, the bradykinin, it really matches, this hypothesis matches the overall COVID symptoms. So vitamin D, the vitamin D receptor, that's downregulated. So anything on this is again from that paper that we're discussing here today, anything that's in blue is downregulated. Anything that's in red is upregulated. This is the simplest picture they've produced of this whole thing. I like it a lot because it matches to the symptoms. So out here in the kidney, which is producing renin, we're seeing a upregulation of renin production, a downregulation of the inhibitory vitamin D function. So having more vitamin D in your system actually can really help in this case. ACE itself is downregulated. ACE2 is upregulated. So because ACE is downregulated, we don't get to the angiotensin II part of the pathway, which would then help to increase blood pressure again. ACE2 instead is highly activated, so you get the angiotensin 1-9 pathway. As well, the AGTR2 is upregulated, and that is kind of cool. That receptor will help sensitize the bradykinin receptors, making them even more reactive. And you got the whole bradykinin pathway over here coming down and finding sensitized receptors. And of course, because we have ACE has been downregulated, we're not eclipsing that pathway. So what happens when all that happens? Well, you get all these things: hypokalemia, arrhythmia, heart failure. Those would all be predicted things that you would see in the heart. You would see vasodilation, hyperpermeability, inflammation, hypotension. And by the way, that hyperpermeability could be related to the hypercoagulability, because once the vessels get damaged and leaky like that, clotting is a thing that very typically can follow. Why? Because the basement membrane of those vessels gets exposed, because when it gets leaky, it doesn't just get leaky. What happens is each tube of your artery or vein is coated, 100% coated by endothelial cells lining the insides of those pipes. When they get leaky, those endothelial cells pull back a little bit or a lot. It creates little holes there. And when that happens, the basement membrane gets exposed. And once the basement membrane's exposed, then all sorts of clotting factors can fire up around that. As well, you would expect pulmonary edema, hyaluronic acid, hydrogels to form. You would expect this encephalopathy, which would be dizziness and headaches, cognitive impairments, and ischemia, and as well the myalgia, the muscle aches and pains and all that. So and particularly the pain that people experience in their muscles. So because the bradykinin pathway also can increase pain. At any rate, it matches. The whole thing matches all the COVID symptoms. So it's very cool piece of work. I like it because it confirms a lot of the stuff that we've been uncovering here for a long time, and it matches a lot of the things that we've been talking about and introducing as concepts based on the other signals we were getting. This is the explanatory function for why this might be happening, but we were still getting perfectly valid, perfectly reasonable results from seeing the improvements that would happen in patients' COVID responses based on their vitamin D levels, based on how much zinc they had in their system, and based on, zinc. Hold on. Skipped right over that part real quick here. Let me back way the heck up here because I don't want to miss that at all. Nope. Where did I put it? Here it is. Yeah, check this out. This kallikrein, this protease here that converts these precursors down into bradykinin itself, it's inactivated by zinc. There you have it. And if the gene is inactivated by zinc, you got to get the zinc somehow inside the cell, because where are genes activated or inactivated? Well, inside the cells. So the zinc is an important component in this whole thing. Can't believe I skipped over that. Drbeen pulled it right out and talked about it. It's also in the paper itself where they're talking about the role of zinc. So sorry I skipped over that before, but let's not right now. Let's make sure we understand zinc. Really important part of this story, because if you can inactivate this with zinc, well, then you don't end up with more of this stuff. And if you can have less of that, then you can help short circuit this part of the pathway. And that would be fantastic, right? So at any rate, as we look through all of this, these COVID symptoms are matched. This is a pretty tight match right there. I like it. I'm not sure, again, that this explains 100% everything. There's only the bradykinin storm. I think there is a cytokine storm as well. There is still a coagulation problem that needs to be dealt with. None of this would say we would do away with the MATH protocol, which is the methylprednisone and the heparin and things like that, because all the bradykinin storm will kick off all sorts of other things that can happen. And remember, these are all overlapping, very highly complicated things. So notice if you come down the bradykinin pathway through the B1, B2 receptors, you do end up with cytokine release and things like that as well, this cytokine release down here. So none of this invalidates how we would do some of the treatments that we've seen pull out earlier, but it does provide additional explanation for why we're seeing these things, and it points to a lot of other potential compounds out there that can directly interfere with the bradykinin pathway and its receptors and block those and inhibit those, which could really help provide some benefit to all of these listed symptoms. All right, summary for today, the bradykinin hypothesis, pretty cool, very cool. Very cool. Matches the symptomology really well. I like that. If the hypothesis bears out, hey, many new treatments are going to become available, because guess what? There's already pre-identified and licensed compounds that will attack this whole pathway side of things over here. As well, the surprise to me, though, was the degree to which SARS-2, it up and down regulates gene expression. Wasn't expecting that. It's not just a little bit, like we saw a twofold increase, hundreds of times increase. Just amazing, very sneaky, honey badger worthy. Oh my God. It's just the level of computational firepower that a virus actually represents in terms of what it's able to do is off the charts. There's just no way to... It's just unbelievably complicated how this actually works. The idea that this virus somehow just naturally erupted with all of these capabilities is not as likely to me as the idea that it was helped along by taking viruses that had worked out this computational nightmare over many, many millions of years of evolution, but then need a little help to actually unlock the keys to get into the cells and begin to do all this more complicated stuff. This hypothesis, the bradykinin hypothesis, explains why vitamin D, why zinc, ivermectin have all floated to the top of my list over time as recommended supplements. The signal was there. Now we have a framework to understand the signals, begins to all click into place. So I'm pretty excited by that. And of course, to help get the zinc into the cell, both hydroxychloroquine and quercetin, those are useful, and ionophores that help get the zinc into the cell. And this is the sort of big data I was advocating for all along. I'm really thrilled that it was done. Super nice work. Be great if this was done back in April or maybe May, even earlier, if the CDC had been and FDA had been up to what they should have been doing. All of that. And by the way, for all the people who designed studies and left zinc out for no good reason, right? Except, I don't know, they were petulant or they didn't like it as a theory because they're conflicted by pharma money, or they hate Trump and somehow it was conflated with hydroxychloroquine. Shame on all those researchers who did that. No reason to have been leaving zinc out this whole time. It obviously no downside and plenty of upside, and it should have been included all the way through. It's something that, of course, I've been on since day one. And, of course, didn't have to be this way. It didn't have to be this way. It really didn't. We could have long time ago, right? How is it that come on, I have a degree in pathology and I'm just sitting here talking to you over a Comcast, connection, and somehow I managed to come up with this idea that what we needed was the data. We needed to collect all this data and sift through it as fast as possible. That was way back in April 23rd. And thank you to the people here, Garvin et al. And all these great people here, Alvarez, Miller, Prates, Walker, Amos, Mast, Justice, Aronow, and Jacobson, for everybody who worked on this and cranked through the massive data to sort of have this hypothesis pop out. Good work. I like it, not just because it confirms everything I've been talking about, but because it makes sense. It clicks. It has that explanatory power where when you run into it, all of a sudden you go, yep, there it is. That fits. And so I'm really pretty sure this hypothesis is going to be borne out. Maybe not, but let's see it all get repeated, and let's now test the hypothesis with some of the bradykinin receptor antagonists and things like that. And as well, let's keep it up with everything that we've recommended about boosting your immune system. So remember, you want your quercetin, you want your zinc, you want your vitamin D. Make sure you're getting those things, of course. And the other lasting shame beyond, the researchers who haven't been, clinical researchers who haven't been including vitamin D and haven't been including zinc and things like that. NIAID, Fauci, all those people, NIH, that have not been advocating vitamin D all the way through, that have not been advocating the various supplements that people should be taking if they happen to have deficient diets, which we know a lot of people do. That would have been a great public health benefit. The fact that they didn't do that, so that, I don't know why, so pharma could make a few more bucks on the next exciting pharma intervention and all of that. Great, that's fine. But, if you really wanted to have, if you really wanted to defund something, it shouldn't be the police in this story. Deunionize them, sure. Demilitarize them, absolutely. But if you want to defund somebody to teach them a lesson, that would be the NIAID. That would be Fauci. That would be the NIH. That would be the places that are fundamentally not doing their job of promoting public health when that is their core mission, ostensibly, right? All right, that's it. That's all I have for today. Remember, get a t-shirt, join the movement. I think there's just really, really bad things coming along economically. I think it's tied to the fact that oil production is way down. In fact, I'm writing about that this week, and I'm talking about oil and oil production. Yes, I'm a multifactorial person, and I'm really intrigued by what's happening economically. I do think there's a double dip recession coming. I do think that there's some real pain along the way, which means we're all going to need to be resilient. And having lots of resilience in your life means doing things for yourself, reskilling. It means not waiting around for somebody to tell you that things are about to get bad. It's getting ready on your own terms. And it's not about putting on a hair shirt. It's not about hunkering down, waiting for another shoe to drop on your head. It's about very proactively thinking about and building towards a regenerative lifestyle. That's what a truly resilient lifestyle is. It's a regenerative lifestyle where your garden soil is improving every single year so that when you leave this earth, you've left it in a better place. That's what the movement is fundamentally about. All right, remember, hey, like this. If you like this, press the like button. That'll help other people find it. As well, you got to remember to resubscribe if necessary, because I bring these things along with my opinions that maybe the NIAID ought to be defunded. Google frowns on that kind of stuff, and YouTube and Twitter and things like that, because I'm not showing my fawning adoration for the big institutions and entities out there. That's not what I do. At any rate, resubscribe if you have to. Hit like if you liked it. If not, don't do it. Or hit the thumbs down if you didn't like it. And have a great weekend. We will see you next time. Thanks for listening. I appreciate it, everybody. Can't wait to see where all this goes. Bye-bye.
Hi, folks. This is Adam Taggart, Chris's co-founder at Peak Prosperity. To get your own Resilience shirt, simply go to peakprosperity.com/shirts. Each shirt is made of 100% organic cotton and produced in the USA. And when your shirt arrives, please send us a photo of you wearing it if you feel comfortable doing so. We'll add you to our wall of proud Peak Prosperity members who are showing the world that resilience is the way to a better future. Thanks for watching.